the principle of the packed cell volume, as we have discussed some of the parts earlier, the value of the test, the materials, the equipment. Now we discuss the principle and test method and some of the quality controls. So basically, if you're talking about the principle of the packed cell volume, you're talking about the proportion, the quantity, the amount, of whole blood that is, a kind of you get, when you spin the sample. You are looking at the red cells, the ratio of red cells and other parts of, the blood components. Like when you spin the sample, you find out that it is being divided into three parts: the plasma, the yellowish part, and you have the buffy coat where the platelets and the white blood cells are, and also the red blood cells. So actually, in the principle, or in this case, what we are aiming, or what you are looking at, is our point of concentration is the red blood cells. So anticoagulated blood, that is being filled into a capillary tube of a specified, millimeter, about 75 millimeter, so which has been covered and spinned, after a period of time, about 3 to 5 minutes. The red blood cells will be packed, below the tube, by whereby the whole blood is being separated, and you use the hematocrit reader to read the result of the test which has been done. So what are the methods? The method we have discussed earlier. I say that we have the automated methods, whereby when you want to run full blood count using the automated hematology analyzers, such as Abacus, we have the Mindray, and so many other hematology analyzers. So basically, on these methods, we are discussing the manual method. So how do you carry out? So whenever possible, perform the test in duplicate, like in two different tubes, to compare the results, even to see the variation, the differences. So what do you do after you get a whole blood, you know, be it inside an EDTA container? Okay, so let's assume you are using the whole blood, that is the blood inside an EDTA container. So what do? You fill, 3/4 of the blood into the tube. Okay, so a plain tube, you understand. When you're using a plain tube, you use an anticoagulated blood, while if you are using a heparinized capillary tube, you use a direct blood that has been collected by either a poncho, you know, from the patient into the tube. So the next thing you do is if you are using the whole blood, to use a tissue, to remove the excess blood from the capillary tube. So the next thing you do is you seal, the unfilled end of the capillary, using a sealant material. A sealant material, like shows we have this. So on the practical, basically we are going to see how it is being done. So after you fill 3/4 of the capillary tube, the next thing you do is you seal it using a sealant. So, but some people, uses phlegm. But in using phlegm, there's this thing you need to be aware of. Do not expose the tube, to the phlegm, because it is going to, lyse, or let me say burn the red cells, which will affect the result, and of which what we are looking at, or what we are aiming at in this process, is the red blood cells. So the next thing what you do is, carefully locate the filled capillary tube after filling it, after sealing it. So you have to locate it into the tube, into the centrifuge, inside the rotor and locate the number. There is a number inside the hematocrit centrifuge, and write it on the form. For example, you're running about 3 to 4, 5, samples. So you may number them 1, 2, 3, 4, 5 on the form, and likewise inside the machine, to avoid errors inside during the process or the work. So the next thing you do is you centrifuge. So for about 5 minutes, for 3 to 5 minutes, for about a kind of 12,000 revolutions to 15,000 revolutions. These machines are able to do that. So immediately after you finish centrifuging it, the next thing you do is to check whether there is no break. Sometimes the capillary tube may break inside the machine, or there is no. You have to check whether there is no spillage of blood. So if there is spillage of blood, you have to use the gloves and clean the machine and redo the test again. So how you do the reading is using the hematocrit reader. So basically that is all about the PAC cell. So in summary, we say the PAC cell is a test that doctors require, before and after surgery. Is a test used for screening anemia, and is also used in investigating, dehydration, burns, and polycythemia, and many other things. And when you have accident, you lost blood, you use the PCV also. So, and some of the materials, we say we have the hematocrit centrifuge, hematocrit reader, we have the capillary tube. Also, we have the sealant or the clay is a part of the materials used. We say you can either use the manual method or the automated method. And the principle, we talk about the proportion of whole blood, the red blood cells. So you compare them and see the results. So basically, this is all about the test. On the next video, we are going to do the practicals and see how it is being done clearly, step by step. Thank you very much.